Welcome, deep divers, to the deep dive. Today, we're diving headfirst into the really fascinating world inside plant cells. Our mission to figure out how these tiny biological machines manage everything coming in and going out and how they talk to their neighbors. We're using a classic text as our guide, a chapter from Raven Biology of Plants, the 8th edition. Because, a cell isn't just sitting there, it's this incredibly active place, constantly pulling in supplies, getting rid of waste. And the plasma membrane, that outer layer, it's the gatekeeper, making all the decisions. We're going to unpack how it all works, from water to big molecules, even how cells communicate.
Absolutely. And understanding that gatekeepers, the membrane, it's fundamental to understanding life. What's really amazing is that these membranes aren't just passive barriers. They actively create and maintain differences in chemical concentration, in electrical charge. And these aren't just, minor details. They represent potential energy, stored energy that powers so much of what the cell does. Without that control, that regulation, life just wouldn't work efficiently. It's one of the key things separating living things from, non-living stuff.
Okay, let's jump right in. If we're talking movement in and out of cells, the most obvious place to start has to be water, right? It's everywhere. So what drives water's movement?
You hit the nail on the head. Water is the main event in many ways. Its movement boils down to three basic processes: bulk flow, diffusion, and osmosis. Bulk flow is maybe the easiest to picture. It's just the overall movement of the liquid because of differences in potential energy. Think of a waterfall. The water at the top has higher potential energy, what we call higher water potential, than the water at the bottom.
Right, it just naturally flows downhill, energetically speaking.
Exactly. Water always moves from higher water potential to lower water potential. And it's not just gravity. Pressure can create water potential too. If you squeeze a hose, you can make water move uphill against gravity if the pressure potential is high enough.
And that's a big deal in plants, isn't it? That bulk flow.
Huge deal. It's the primary way sap sugar dissolved in water moves long distances in the phloem, from the leaves down to the roots or fruits, sometimes over many, many meters.
Okay. And scientists can actually measure this potential.
Yep. They measure water potential in units called pascals, or usually megapascals, MPa. Pure water is the standard, set as 0 MPa. Adding solutes, like sugar or salt, makes the water potential negative. It lowers it. So this measurement lets plant scientists predict which way water will move. It's like a weather map for water movement in and around cells.
Okay, that makes sense for water moving as a whole liquid. But what about the tiny individual movements? Diffusion. That sounds more random.
It is random on the individual molecule level. Diffusion is just the tendency of substances to spread out due to the random motion of their ions or molecules. Think about dropping dye in water or spraying perfume. The molecules themselves are just bouncing around randomly, but the net result, because there are more in one spot initially, is they move from high concentration to low concentration, down the concentration gradient.
So no tiny GPS needed, just probability pushing them outwards until they're evenly spread.
Exactly. Until equilibrium is reached, where there's no net movement anymore, though individual molecules are still zipping around. And diffusion is faster in gases than liquids and faster when it's warmer. This applies to water, too. Where you have lots of solutes, you have less water concentration, which means lower, more negative water potential. So water diffuses down its own concentration gradient, which is the same as saying it moves from higher to lower water potential.
Okay, but how does this random spreading work with the cell membrane in the way? Doesn't that block things?
It does, but selectively. Small nonpolar molecules, things like oxygen or carbon dioxide, they slip through the lipid part of the membrane pretty easily. And even small, uncharged polar molecules like water itself can sneak through momentary gaps. For other small polar molecules, it's a bit like a sieve. The smaller you are, the better your chance of diffusing across. But the membrane is definitely a barrier for larger or charged stuff, relying only on diffusion.
And diffusion is slow over distance, right? So how do cells cope?
Good point. It's really slow over anything more than microscopic distances. Cells have a couple of tricks. One, they keep concentration gradients steep through metabolism, like they constantly use up oxygen inside, so there's always a strong pull for more oxygen to diffuse in, or they produce CO2, creating a gradient pushing it out. Two, inside the cell, they use cytoplasmic streaming. The cytoplasm actively flows and swirls around, mixing things internally much faster than diffusion alone could manage.
Ah, clever workaround. So diffusion handles some things, but what about when water needs to move more purposefully? You mentioned osmosis.
Right, osmosis. This is specifically the movement of water across a selectively permeable membrane. That membrane lets water pass, but blocks other things like sugars or salts. And the rule is the same. Net water movement is from higher water potential to lower water potential, which usually means water moves from an area with fewer solids to an area with more solids.
So it's like water is drawn towards the dissolved stuff if it can't pass through the membrane itself. How can we visualize that?
The classic experiment, described in the book, uses a tube with a membrane at the bottom, filled with sugar solution and dipped in pure water. Water rushes into the tube, trying to dilute the sugar. The water level in the tube rises until the pressure from that column of water physically pushes back and stops more water from entering. The pressure needed to stop that net movement, that's called osmotic pressure. And the tendency of the solutes to draw water in is related to osmotic potential, which is always a negative value.
Okay, that makes the force clear. What does this mean for actual living cells? Sounds like it could cause problems, like cells bursting or shrinking.
It absolutely presents challenges and opportunities. Think about a single-celled organism like Euglena living in fresh water. Its insides have more soups than the pond, so water constantly rushes in by osmosis. To avoid bursting, it has a contractile vacuole, basically a tiny pump that constantly collects and squirts out excess water.
A little cellular sump pump. And plants, they have rigid walls so they don't burst, right?
Right. Plant cells use osmosis to their advantage. They actively pump solutes into their large central vacuole, making the inside have a much lower water potential than the outside soil water. Water flows in via osmosis. But instead of bursting, the cell membrane, the protoplast, pushes against that strong cell wall. This creates internal pressure called turgor pressure. It's what makes plant cells firm or turgid. It's literally what holds up leaves and nonwoody stems. The wall pushes back with wall pressure, creating a stable, pressurized state.
So that's why plants wilt. They lose turgor pressure.
Precisely. If you put a plant cell in a salty or sugary solution, something with lower water potential than the cell, water will leave the cell by osmosis. The protoplast shrinks and pulls away from the cell wall. That process is called plasmolysis. You can see it happening in Elodea leaf cells under a microscope. It leads directly to wilting.
Okay. And there was one other water thing mentioned. Imbibition.
Ah, yes, imbibition. It's related to water's properties, how water molecules stick together, cohesion, and stick to surfaces, adhesion. Imbibition is when water moves into absorbent substances like dry wood or gelatin, or importantly, a dry seed. The water gets absorbed into the matrix, causing it to swell, and this can generate huge pressures. It's absolutely crucial for seed germination that initial swelling helps break the seed coat.
Fascinating stuff. Water. Okay, so we know what moves, especially water. Let's zoom in on the structure that controls it all, the membrane itself. What's the key takeaway about its structure?
I think the key is its dynamic and complex nature. The basic framework is the lipid bilayer. 2 layers of lipid molecules, mainly phospholipids, and in plants, sterols like stigmasterol, like cholesterol, like in animals. Embedded within or attached to this bilayer are proteins, lots of them. Many are transmembrane proteins that span the entire width, with parts sticking out on either side. These proteins often have hydrophobic sections tucked into the oily lipid core and hydrophilic sections exposed to the watery environment.
Like little machines embedded in a fatty wall?
Kind of, yeah. And you have integral proteins that are really tightly embedded, and peripheral proteins more loosely attached to the surface. The classic model, the fluid-mosaic model, described this as lipids and proteins floating around laterally, like icebergs in a sea.
But that model's evolved a bit.
It has. We now think it's maybe less uniformly fluid. The membrane might have variable thickness, a higher proportion of proteins than first thought, and importantly, proteins might be organized into specific functional groups or complexes, not just floating randomly. And lipids might form distinct patches too. It's more organized, more like a dynamic cityscape than a simple sea.
And there are things on the outside surface too.
Yes, on the outer face of the plasma membrane, you find short carbohydrate chains, oligosaccharides, attached to proteins forming glycoproteins, or attached to lipids forming glycolipids. These act like cell identity markers crucial for recognition, like how cells respond to hormones or interact with microbes.
So the lipids provide the basic barrier, but the proteins are doing most of the specific jobs.
That's the gist. The lipid bilayer gives structure and basic impermeability, especially to charged things. But the proteins act as enzymes, as carriers for transport, as receptors for signals. They handle the specialized functions. Membranes that do a lot of energy work, like in mitochondria or chloroplasts, are packed even more densely with proteins.
Okay, that paints a clear picture of the gatekeeper structure. Now, how does it actually let the right things through? We know small, nonpolar stuff slips by, but what about essential polar molecules or ions?
That's where the transport proteins we just mentioned come into play. They provide specific pathways. They're highly selective. A protein that transports potassium ions usually won't transport sodium ions, even though they're similar. They create a safe passage, avoiding the hostile hydrophobic lipid environment. We can group these transporters into roughly 3 classes based on how they work and how fast they are. First, you have pumps. These use energy, usually from ATP, sometimes light, to actively move substances against their gradient. Think uphill. They are relatively slow, maybe less than 500 molecules per second. The proton pump, H plus ATPase, is a classic plant example.
Okay, slow but powerful. What's next?
Carriers. These bind to a specific molecule, then change shape, kind of like a revolving door, to shuttle it across the membrane. They don't usually require direct energy input if moving down a gradient, but they're faster than pumps, maybe 500 to 10,000 per second.
Faster. And the fastest.
Those are the channels. These form pores, like tiny tunnels, through the membrane. When the channel is open, specific ions like sodium, potassium, calcium chloride can just flow through very rapidly down their electrochemical gradient. They're usually gated, meaning they open and close in response to specific signals. And their speed is amazing, tens of thousands to millions of ions per second.
Millions per second. That's incredible flow.
It really is.
Yeah.
And interestingly, even water, which can diffuse across the lipid bilayer slowly, gets a major speed boost from specialized water channels called aquaporins.
Ah, so water gets its own express lane.
Exactly. Found in the plasma membrane and the vacuole membrane, the tonoplast, they allow for much faster water movement than simple diffusion. This is vital for things like rapid water uptake by roots, movement through the xylem during transpiration, and sometimes even blocking water entry if roots get flooded. They're dynamic regulators.
So much traffic. Now, you mentioned moving uphill versus downhill and energy. Let's unpack passive versus active transport.
Right. It all comes down to gradients. For uncharged molecules, it's simple. Movement is determined by the concentration gradient. High to low is downhill. But for charged ions, it's 2 factors: the concentration gradient and the electrical gradient, or voltage, across the membrane. Together, that's the electrochemical gradient. Plant cells are typically electrically negative on the inside compared to the outside.
Okay, so ions feel both a chemical push and an electrical pull.
Precisely. Now, passive transport is movement down the concentration or electrochemical gradient. It doesn't cost the cell direct energy. Simple diffusion is passive. So is facilitated diffusion, where a carrier or channel protein helps the substance move downhill.
And facilitated diffusion can be specific.
Very specific. We see uniport, where one substance moves one way. That's how channels work. And then there's co-transport, where the movement of one substance is coupled to the movement of another. Co-transport can be symport, where both substances move in the same direction across the membrane, or antiport, where they move in opposite directions, like trading one thing for another.
Okay, that's passive, the coasting downhill option. What about active transport?
Active transport is the uphill climb. It's moving a substance against its concentration or electrochemical gradient. This always requires the cell to expend energy, and it's always mediated by carrier proteins, often called pumps.
Where does the energy come from?
2 main ways. In primary active transport, the energy comes directly from ATP hydrolysis, or sometimes light. The plant proton pump, H plus ATPase, is the prime example. It uses ATP to pump protons, H plus, out of the cell, building up both an electrical potential and a pH gradient across the membrane.
So it's creating a form of stored energy.
Exactly. And that stored energy is then used in secondary active transport. The protons that were pumped out want to flow back in, down their electrochemical gradient. As they flow back through a different carrier protein, that protein uses the energy of the proton flow to drag another molecule, like sucrose, against its gradient into the cell.
Wow, that's clever. Use energy once to create a gradient, then use the gradient to power other transport, like a rechargeable battery system.
A very good analogy. It allows plant cells to accumulate sugars and other essential nutrients to concentrations much higher than outside the cell.
And scientists can actually study these tiny protein machines individually.
Yeah, there's an incredible technique called patch clamping, mentioned in a sidebar in the text. Researchers use a microscopic glass pipette to form an ultra-tight seal on a tiny patch of cell membrane, sometimes just isolating a single channel protein. They can then measure the electrical current as ions flow through that one channel, and even change the solutions on either side to see how the channel responds. It gives amazing insight into how these transporters work.
Incredible precision. Okay, we've covered water, ions, small molecules, but what about the really big stuff, like huge protein molecules or even whole bacteria? Transport proteins can't handle those, right?
No way. For the heavy-duty cargo, cells use vesicle-mediated transport. This involves creating little membrane-bound sacs called vesicles that either bud off from or fuse with the plasma membrane.
So like using little delivery trucks made of membrane.
Pretty much. Transport out of the cell is exocytosis. Vesicles, usually originating from the Golgi apparatus, travel to the plasma membrane, fuse with it, and release their contents outside. This is how plant cells secrete components for the cell wall, like hemicelluloses and pectins, or the slimy mucilage that lubricates root tips. Carnivorous plants even use it to release digestive enzymes.
Okay, that's shipping things out. What about bringing things in?
That's endocytosis. The plasma membrane dimples inward, enclosing material from the outside, and then pinches off to form a vesicle inside the cell. There are a few types. Phagocytosis is cell-eating, engulfing large solid particles like bacteria or cell debris. It's common in amoebas, but in plants it's quite rare, though a key example is how lagoon root cells take in rhizobium bacteria. Pinocytosis is cell drinking, taking in droplets of external fluid and dissolved solutes. It's basically the same mechanism, but for liquids, and it happens in pretty much all eukaryotic cells. And then there's receptor-mediated endocytosis, which is highly specific. Molecules outside bind to specific receptor proteins clustered in areas called coated pits on the membrane surface. These pits, often coated with a protein called clathrin, then invaginate and pinch off, forming coated vesicles carrying the specific cargo and its receptors into the cell. It's a way to import specific needed substances very efficiently.
And the cell reuses the vesicle membrane parts, right? It's not wasteful.
Absolutely. There's constant recycling of membrane lipids and proteins, including those receptors, between the plasma membrane and internal compartments like the Golgi during both exocytosis and endocytosis. It's a very dynamic and efficient system.
Right. Okay, we've got a good handle on how individual cells manage their borders. But plants are multicellular. How do all these cells coordinate? How do they talk to each other?
Great question, because they absolutely have to communicate to function as a whole organism. There are two main ways this happens in plants. First, there's signal transduction. It's how cells respond to chemical messengers, like plant hormones. A signal molecule produced elsewhere travels to a target cell.
Like receiving a message.
Exactly. It typically involves three steps. Reception. The signal molecule, the first messenger, binds to a specific receptor protein, usually on the cell surface. Transduction. This binding triggers a cascade of events inside the cell, often involving the generation of second messengers, small molecules or ions that amplify the signal. Calcium ions, K2+, are a super important second messenger in plants, often released from internal stores. Induction. These second messengers, maybe Se2 plus binding to a protein called calmodulin, then activate specific cellular processes, like turning on enzymes, leading to the cell's response.
So receive the signal, amplify and translate it, then carry out the instruction.
You got it. It's a way for cells to respond specifically to external cues or messages from other parts of the plant.
And the second way they communicate sounds more direct.
It is much more direct. These are Plasmodesmata. They are narrow channels of cytoplasm that pass directly through the cell walls, connecting the living contents, the protoplasts, of adjacent plant cells.
Wow, actual physical bridges between cells?
Exactly. All the interconnected protoplasts form a continuous network called the symplast. Moving through these connections is symplastic transport. Moving through the cell walls outside the protoplasts is apoplastic transport. Plasmodesmata mostly form during cell division when bits of the endoplasmic reticulum get trapped across the new cell wall. But secondary ones can form later, connecting cells that weren't originally sisters. Structurally, each is a plasma membrane-lined tube containing a modified strand of ER called the desmotubule. Transport mainly happens in the little gap, the cytoplasmic sleeve, around this desmotubule.
So substances can just flow from cell to cell cytoplasm without crossing a membrane?
Essentially, yes, for small enough molecules. It's a much more efficient pathway than having to cross two plasma membranes in a cell wall every time. It's vital for distributing nutrients and signals throughout plant tissues, especially connecting the vascular tissues, xylem and phloem, to surrounding cells.
And we know things actually move through them.
Oh yeah. Experiments using fluorescent dyes that can't cross membranes show it clearly. Inject the dye into one cell, and you can watch it spread into neighboring cells directly through the Plasmodesmata. They are selective, though. Most allow molecules up to about 800 or 1,000 Daltons, so sugars, amino acids, small signaling molecules can pass. But the permeability can vary, creating distinct symplastic domains within the plant. They can even transmit electrical signals between cells.
Are they just passive tunnels?
We used to think so, but not anymore. They are dynamic structures. They can open or close, change their size exclusion limit, and even traffic larger molecules like specific proteins and RNA molecules, which plays a huge role in coordinating development. Things like callus deposition or the cell's internal cytoskeleton seem to be involved in regulating their function. So wrapping up this deep dive, we've journeyed through the incredible ways plant cells manage their boundaries. They are truly masters of selective permeability, using everything from simple diffusion and the power of osmosis to highly specific energy-driven pumps and carriers, and even large-scale vesicle transport for bulk cargo. It's a really sophisticated system for controlling that internal world.
Yeah, and we've also seen they aren't just isolated units. They're part of this larger interconnected community, constantly communicating through chemical signals via signal transduction. And through those direct cytoplasmic links, the Plasmodesmata, it's amazing coordination.
It really is. The key takeaway is that the plasma membrane isn't just a wall. It's this incredibly dynamic, intelligent interface. Water movement, driven by water potential, is fundamental. Solute movement is tightly regulated, often by intricate protein machinery, and all this transported communication allows the plant to function as one cohesive, responsive organism.
Absolutely. It makes you think, doesn't it? When you consider how these microscopic cells manage all this complex traffic and signaling, what does it tell you about how plants sense and react to their environment? Is there one central controller calling the shots, or is it more like a vast, decentralized network where each cell plays its part based on local cues? Something to chew on next time you're looking closely at a plant. Thanks for joining us for this deep dive into the bustling world of the plant cell membrane. We hope it gave you a new appreciation for the hidden complexity of plant life.